everyone, my name is Emily Truchon and welcome to Crash Course with Ethan Thompson, Pierce Strayer, and I. Today we will be learning about phytochemicals. If you're like me, growing up, your mom always said, eat your fruits and vegetables. My stubborn self would always ask why. She just said because they're good for you. Now I know that eating fruits and vegetables are good for you because of the vitamins and minerals they contain. But is there more to it than that? Recent research showed that the benefits of eating plants goes beyond just vitamins and minerals due to phytochemicals contained in the plants. But what are phytochemicals? Phytochemicals stem from the Greek word phyton, which means plant. So phytochemicals literally means plant chemicals.
All right, Emily, thank you for the introduction. And as you mentioned, what we're going to be talking about today are phytochemicals. A recent article came out on Harvard Health, their website, speaking about just how beneficial these compounds can be for humans. Uh, Phytochemicals are responsible for things such as the color of carrots or the spice that you get in a jalapeno. They're secondary plant metabolites that are made primarily to protect against insects and convey better survivability to the plant. However, they also can have beneficial medicinal effects on the people that eat them. So you're not just getting the benefit of the vitamins and minerals every time you eat a fruit or vegetable, but you're also gaining the effect of whatever phytochemicals may be in it. These phytochemicals range from things such as proanthocyanins to carotenoids and flavonoids, which can have effects ranging from anti-cancer to longevity and even boosting immune system in the case of something like allicin that's naturally produced by garlic. Um, there's an estimated over 5,000 phytochemicals being produced by plants, and that's honestly a low estimate as research is done in the area. We can find out just how many of those are medicinally active, and research on that can expand. This is a very important topic in the pharmaceutical industry, as over 40% of drugs currently on the market are plant derived. And as you can see from some of the articles that we're going to go more in depth on, uh, there's a lot of applications being currently looked into with these substances. All right, here you go, Ethan.
Thanks, Pierce. One of the claims of the news article is that flavonoids in berries, apples, citrus, onions, soybeans, and coffee may fight inflammation and tumor growth. Chrysin is a type of flavonoid that has been well documented to have anti-inflammatory and in recent studies shown effective in reducing tumor growth. These researchers pose the question, since chrysin has been shown to have anti-cancer properties with low toxicity, could it be used to treat melanoma in mice to open the door for future melanoma treatments within humans? In order to test this hypothesis, the researchers used mice as a model for the human body. Each mouse was injected with melanoma cells to create equivalent tumors to test across the groups. The two groups that were used were mice given corn oil every 2 days or mice given corn oil and chrysin every 2 days. The researchers had two trials with this method, one that lasted for 2 weeks and another that went on for 3. Once these trials were over, the researchers removed and inspected the tumors and compared them against one another. They found that the mice that were treated with chrysin had reduced tumor size compared to the ones that did not receive the treatment. These findings allow for further investigation into chrysin anti-tumor properties within humans, which is more difficult to describe within mice, but would serve as an excellent tool to help treat individuals with cancer.
Following up on the evidence Ethan just shared with us, another study investigated the potential of phytochemicals found in Allium wallacei in the prevention and treatment of cancers. Allium wallacei, more commonly known as jimber, is a Nepal native plant. This was the first study of jimber in this context. However, many other similar Nepal native plants have been found to have cytotoxic effects against different cancer cell lines, so it seemed like a pretty promising candidate. Before we get too into the study, cytotoxic means toxic to cells. So cytotoxic effects against cancer cells is referring to these phytochemicals having destructive effects on cancer cells. Now, back to the study. The researchers hypothesized that jimber could be used as a promising anti-cancer agent with its phytochemicals acting as a cytotoxic against breast, cervical, prostate, and Burkitt's lymphoma, or B lymphoma cell lines. Their experiment was performed using the dried plant extracted in aqueous ethanol and performing phytochemical screening, antimicrobial assay, antioxidant assay, cytotoxicity assay, and flow cytometric analysis. These tests detected the presence of these phytochemicals: steroids, terpenoids, flavonoids, reducing sugars, and glycosides in the plant extract, and the plant showed moderate antimicrobial and antioxidant activity. More importantly, the plant demonstrated moderate cytotoxicity against prostate cancer, cervical cancer, and breast cancer, and good toxicity against B lymphoma cells. These results account for the fact that Jimber, together with its moderate antioxidant and cytotoxic activity and the presence of the phytochemicals, can be a very important candidate to be used as an anti-cancer agent. Moreover, since the crude extract showed promising cytotoxicity, we can conclude that the separation of the plant parts and isolation of the pure bioactive compounds from Jimber can have even better activity against cancer. Therefore, more data is needed in this aspect.
As we saw in the studies presented by Ethan and Emily, phytochemicals have plenty of medicinal applications, and the research being done on them was quite extensive. There was a paper that recently came out in 2018 that focused on the phytochemical asiatic acid that's produced by Indian pennywort and its potential as a glaucoma treatment. The researchers in this study created rat models of glaucoma and administered intravitreal injections of asiatic acid to these rats and observed the results. They were able to use immunofluorescent protein labeling assays in order to determine the function of retinal ganglion cells, as well as the rate of death amongst those cells. What the researchers were able to determine was a very beneficial outcome, with there being decreased levels of cell death and increased levels of those cell functions due to asiatic acid's ability to regulate pro and anti-apoptotic factors through impacting the body's natural signaling pathways, further showing and demonstrating the ability of phytochemicals to impact mammalian and potentially human health.
The news article from Harvard Health Publishing, as suspected, was correct with its claims and is supported by peer-reviewed papers. We discussed a few papers that support this article, such as Christin suppressing melanoma in mice, asiatic acid preventing retinal ganglion cell apoptosis within mice, and antimicrobial, antioxidant, anti-cancer properties of a plant from Nepal. Hopefully now your understanding of how impactful phytochemicals are and their usefulness in both everyday life and within complex medical application has increased. To make sure that you are listening, here's a question that we'd like to ask you. Plant secondary metabolites can be useful in treating this discussed type of human disease that is difficult to treat. A. Influenza. B. SARS-CoV-2 or coronavirus. C. Cancer. Or D. Autoimmune disease. Leave your answer down below. Anyways, thank you for watching this video, and don't forget to like, favorite, subscribe, and we'll see you in the next episode.